This is a recording specifically for RCBC students, but we hope others will join us. We're picking up on Chapter 19, Virology, and we discussed in the last video about how viruses work and what they look like. And now we're going to take a look at a few viruses in particular. Of course, the one that concerns us the most right now is Sars Coronavirus 2. This virus is an RNA virus. Its genes are made up of RNA. It also is an enveloped virus. It has this phospholipid bilayer on the outside. Through that bilayer, studding the bilayer are different proteins like hemagglutinin and this spike glycoprotein, S protein. Remember that glycoprotein means that it's part amino acid and part sugars. When we think about viruses, we want to remember that they're very unique. Each one is very unique. Whether it's made up of RNA, DNA, double-stranded, single-stranded, an envelope around it, they all kind of wreak their havoc in different ways. And one of the things that we want to understand about viruses is what we call their life cycle. This is a life cycle here of coronavirus 2, and what we can see in this gray area here is a cell that the virus is infecting. In particular, the cells have this receptor ACE2, angiotensin converting enzyme 2. That receptor is what the virus uses to gain entry to the cell. This little green protein here is the spike protein, and that spike protein will interact with the ACE receptor, and then the cell will take in the virus. The virus will shed its proteins and uncoat itself so that now the RNA is available. That RNA is going to be used for two things. One, it's going to be translated so that we can get proteins that are needed to make more viruses. And the other thing is that it's going to replicate so that we have more copies of RNA that is going to be used to put inside the new viral particles. So you can see here that new viruses have assembled from the processes outlined here, and then they will leave the cell. As you can see, one virus comes in. In this depiction, it just shows two viruses coming out, but that varies. It can be 100, 1,000, 1,000,000, depending on the type of virus it is. Now let's talk about the damage that coronavirus causes. In particular, we're going to talk about the lungs because that's where we're seeing the primary symptoms. But I do want to point out that this ACE receptor is on different cell types, not only in your nasal tract or respiratory system, but also in certain brain cells, in intestinal cells. So there is potential that some of the symptoms of infection will impact other systems within our body, not just the respiratory system. This is a picture of our lungs. We, of course, breathe in through our nose and our mouth. The air goes through the trachea and then the bronchi. We see the branching here. And then eventually it branches further into bronchioles. And that's kind of what we're seeing here is the terminus of a bronchiole. And that bronchiole then ends in what we call alveoli, or singularly alveolus. And these are just tiny sacs which are on the outside coated with blood vessels, as we can see here. These are very thin sacs so that the oxygen, let's see here, this is the oxygen that we just breathed in inside of an alveoli, can actually move out of the alveoli and then hop onto or into a capillary and within a red blood cell with hemoglobin. And of course, CO2 jumps off and we breathe the CO2 out as a waste product of respiration. So that's what's supposed to happen. What happens with coronavirus is that we'll get a fluid buildup in these sacs. And as we might imagine, if the fluid is there, the transfer of oxygen cannot happen, or it doesn't happen as efficiently. As with a lot of infectious agents, it is often our immune system's response which is causing the damage. If you can take a look in here again, we have the alveoli or alveolus and the capillary. In addition to those red blood cells, the blood in this capillary also has immune cells like macrophages and other neutrophils, right, which we might see right here. These cells sense that there is an infection, and they come in to try to take care of that infection. And as sort of collateral damage, they kill the cells that have been infected by the virus. And so this fluid here that's built up in part is the cytoplasm that's kind of spilled out from inside the cells. There are other fluids as well, but that is one thing that happens as a result of the infection, the infected cell being attacked by your immune system. Now, viruses also, let's move on to another type of RNA virus. Some viruses are latent, so they can infect us, and instead of seeing the results in 2, 5, 14 days, they will actually take their genetic material and integrate it into our host genome, into our chromosomes. And HIV is an example of one of those RNA viruses. Another example, but they do it differently, are herpes viruses. A type of herpes virus is chickenpox. You might know that if somebody has herpes and then they're out in the sun, they might get a flare-up of a cold sore. And chickenpox also can remain latent, and later we see it as shingles as well. And that is a type of herpes virus. So they can kind of go in there, stay stealth for a while, and then cause damage later. Let's specifically look at HIV. HIV is, as I mentioned, an RNA virus. It has capsid proteins around it. It is also enveloped. It has a phospholipid bilayer. And the protein that's on this is also a glycoprotein, but we call it GP120. Another thing about this virus, and it has to do with its latency, is that it is called a retrovirus. So it's RNA, but additionally is a retrovirus. And I will talk about what that means, but as because it's a retrovirus, the virus needs to bring in an enzyme called reverse transcriptase. And we will show you what that looks like when we take a look at the life cycle. Human immunodeficiency virus is the cause of AIDS, acquired immunodeficiency syndrome, spread primarily from sexual contact. It is a blood-borne pathogen, so if you mix contaminated or infected blood with someone who's not, there's the potential that HIV is spread to that other individual. This has become a chronic infection, although when it first came to light in the 80s, it was really a death sentence. But let's not forget that give you some statistics, in 2016, there 3 million adults and children will become infected or became infected with HIV, and 30 million people were living with it worldwide. But 2 million in that year died of AIDS. In the US, about 50,000 people die each year from HIV infections. So this is definitely a very scary and damaging virus. We'll talk about why it is wreaks its havoc. This shows us the global impact of AIDS, And in the very darkest are the highest percentage of HIV positive people. And specifically, you can see here that we have very high rates in Africa, in Southern Africa. And for instance, in Botswana, 23% of the population is HIV positive. Now, why does it have such damage? The reason is that HIV infects your immune cells. It infects specifically T cells or T lymphocytes. Your immune cells are responsible for fighting off foreign pathogens, even internal pathogens like cancer cells, but bacterial cells are fought off by your immune cells. So when HIV infects these cells and these cells die, then you don't have any sort of army to fight off other things that may attack you. Let's take a look at the life cycle here. This is a T cell, this entire region here. The T cell has an important receptor on it called Cd4. Not shown in this picture is also another receptor called CCR5. I will show you that in another graphic. The Cd4 receptor is necessary for HIV to enter the cell. HIV, we mentioned, has GP120 on its surface, and that GP120 has to interact with CD4. When that happens, the genetic material, the RNA of HIV, is able to get inside the T cell. Now, this is where this retrovirus part comes to play. That virus is RNA, the genetic material is RNA, but the virus's goal is to get inside a human chromosome. Our chromosomes are made up of DNA. So the virus needs to make a copy of DNA from the template of its RNA and then make another copy of that so that it ends up with double-stranded DNA that matches the sequence that was up here in the virus. In order to do that, it needs this enzyme called reverse transcriptase that I have abbreviated here, also called RNA-dependent DNA polymerase. The next thing that it does is it takes this DNA, it moves into it tries to put that DNA into the chromosome, and it uses an enzyme called integrase. So now we can see this little portion here is the DNA that was here, that was copied from the RNA that was here, that came from the virus that is here. When something stimulates this cell to come out of latency, the virus will then begin doing a couple of things. It'll cause transcription and translation, which makes viral proteins, but also it will cause transcription, which will make viral RNA. So now the virus's RNA and proteins are ready to be assembled, reconstructed here, and you can see that they're budding off of the T cell. Now, what I want to mention here before we move on to the last slide is that the virus itself, most viruses are very, very simple, and they bring what they need to bring, but they steal everything else from the host. If you remember in the structure of HIV, there was reverse transcriptase brought in as part of the virus. And that is because we do not have, humans, the host, do not have reverse transcriptase. We don't ever go from RNA to DNA. And so the virus needed to bring that virus or that protein, that enzyme with it. Now, in the next lecture, we're going to talk about how to treat HIV. Thank you very much, and I hope you join us then.